Yeah. So, good morning, all of you. This is Dr. Narkade Gopal Vasudev, working as Assistant Professor and Head, Department of Genetics and Plant Breeding, School of Agriculture, SRN University, Baranta. So, welcome to all. Today, we are dealing with the most important topic of hybridization. This plant breeding will start with the process of hybridization. So in this class, we will deal with what are the objective, what is the aim of hybridization, what are its objectives, how we have to proceed in hybridization, what are the different criteria while selection of plants, and what are the different types of hybridizations and their protocols. Okay, so let's start with the topic. So here, as we can see, this parental line A cross with parental line B for the production of F1 hybrid. This is one of the activity, or you can say one of the objective of hybridization for the production of F1. So for the production of F1 again, is there are different criteria for selection of parents? Is there are some criteria for designation of your parent as a male and female? Is there any methodologies for emasculation? After harvesting of F1 seed, can we release that F1 seed directly to the farmer as a cultivar or as a hybrid? Or for improvement of some cultivar, can we use this F1 hybrid? Can we advance this F1 population and can we create some amount of variation so that we can initiate some breeding program with respect to some desirable traits like disease resistant or quality resistant trait? So all these things we will going to see in this class. So first we will start with entry. What is meant by exactly what is meant by plant hybridization? So plant hybridization is nothing but you have to cross between two diverse plants. Diverse means in this case, the plants, two different plants having morphological phenotypical differences, such as like as you can see in this picture, given in P2 are the two parents having morphological differences and height differences also. So likewise, two diverse plants you have to select for crossing purpose with variation. That is called as plant hybridization. After selection of two parents, you have to designate the two parents as a female parent and one as a male parent. After designation, you have to collect pollens from your male parent and you have to dust them on stigma of flower of female parent. So that will complete your pollination, emasculation and pollination process. So after emasculation and pollination process, you will get seeds. So after a successful harvesting of the seeds, that seeds you can call as F1 hybrid seeds. And the generation you will raise from that hybrid seed, you can call it as F1 generation. F1 means first filial generation. Okay. You can see in this picture, in maize, P1 cross with P2, you will get this B. B is nothing but hybrid. So you can see the performance of hybrid is far much better than both the parents. So this superiority of this hybrid over both the parents is called as hybrid vigor. Hybrid vigor is nothing but your heterosis. Okay. Yeah. So we will continue towards next step. What is the chief objective of hybridization? So to start a particular breeding program, we need a population with variation. Population of particular specific crop with a ample amount of variation, and that ample amount of variation we can create by following this hybridization. You can see in the slide, if we cross both parents, we will get F1, and after selfing of this F1, we will get F2 population. F2 population will having segregation. Segregation means we can find some either related to one parent, some with relating to another parent, and some recombinants we will find. So we have to cross the two genetically distinct, genotypically distinct, that is different plants. So what it will happen? So both different genes from both the parents will combine brought together in F1. Okay. So as we all know, F2 is the most segregating population of breeding, segregating population in breeding life cycle. So degree of variation, or else you can say degree of segregation produced in F2 is depends on number of heterozygous genes present in F1. So means if the number of heterozygous genes are more in F1, you will get more amount of segregation or more amount of variation in F2 population. So it's all is depend on how many heterozygous genes are present in F1. So before proceeding, we will see some historical things regarding plant hybridization. So this plant hybridization started in 1717. Started by Thomas Fairchild, who developed first interspecific hybrid, globally known as Fairchild's Mule. First interspecific hybrid between Sweet William and Carnation species of Dianthus. So after that, in between 1800 to 1835, from England, one scientist was there, Dr. T.A. Knight, who first used artificial hybridization techniques in fruit crops. Then in 1866, the great Gregor John Mendel from Austria, who published his discoveries in experiment in plant hybridization, regarding formulation of laws of inheritance from his work on garden pea. So later, he is now known as the father of genetics because of his laws of inheritance. Again, in 1890, the triticale came. Triticale came from the cross between bread wheat and rye. So it was done by Rimpu. He came from Sweden. So first commercial maize hybrid was developed by Jones in 1917. So first commercial hybrid in maize developed by Jones in 1917. So in 1927, Karpenchenko was there who developed first intergeneric hybrid between radish and cabbage in Russia. So now, what are the objectives of hybridization? We have seen the chief objective of hybridization is to create ample amount of variation in that particular breeding population of specific crop. Okay, so what are the different objectives of hybridization? So first and foremost objective is combination breeding. So as itself name indicate, combination breeding. So combination breeding is nothing but transfer of one or more characters from other varieties to a particular variety. So as the main role of breeder as far as crop improvement is concerned, to transfer one or more characters from one variety to other. So hybridization is for the main objective is if we have to transfer one or more characters from different sources into a particular variety. So that process is called as combination breeding, and these characters may be governed by single gene, polygene. For example, like disease resistance and quality traits. So these traits are governed by some by single gene, some by one or more genes. So second is transgressive breeding, or you can say as a transgressive segregation. So as we all know, F2 is most segregating generation in breeding cycle. So transgressive segregation. So what is meant by transgressive segregation? Some segregations we can see in F2 generation some segregations of characters beyond the parental limits, beyond the original parental limits. That is, I'm not saying about beyond F1. So transgressive segregation is the segregation of characters beyond original parental limits. That is P1 and P2 in the segregating generations like F2. And the third objective is production of hybrids. So hybrids, as I already told you, hybrid shows high vigor and yield when we compare that hybrid with parents. So this phenomenon is called as hybrid vigor or heterosis. So you can raise this F1 seed either in bulk or you can distribute directly to the farmers, especially in cross-pollinated crops. So next now we will move towards types of hybridization. So based on these genetic differences between the parents, hybridization can be classified into two types mainly. First one is intervarietal hybridization and second is distant or wide hybridization. So first is intervarietal hybridization, the cross between the members of same species, that is the cross between within the species is called as intervarietal hybridization. In this type of hybridization, there are different types of cross patterns you can see. So first is simple cross. So in this cross, so in simple cross, it is straight cross, that is parent A cross with parent B to cross to yield F1 hybrid. So second type is complex cross. So in complex cross, here more than two parents are involved. So such crosses are called as convergent crosses. Since by following these complex crosses, we are bringing different genes together. We are transferring different genes from different source in a particular variety. So there are again two types for complex crosses, three-way cross and double cross. So what is three-way cross? So three-way cross is nothing but cross of hybrid with open pollinated variety, or simply it is cross of F1 with a variety. So second one is double cross. Double cross means when you will cross two different F1s, crossing of two different F1s, that will gives you a double cross progeny. Okay. So second type is wide hybridization. So wide hybridization. Wide hybridization is what is meant by wide hybridization? So hybridization between individuals from different species. Hybridization between individuals from different species belonging to same genus or between crossing between individuals from two different genera. Okay, so it is of two types. First one is interspecific. And what is interspecific? Crossing between two plants from two distinct genus. Crossing between two individuals from two distinct species of same genus are crossed together. So for example, Oryza sativa cross with Oryza perennis. Then second type is intergeneric cross. When two individuals being crossed belonging to two different genera. So what is interspecific? Cross between two individuals belonging to same genus, different species. And what is intergeneric? Plants belonging to two different genera. So example for intergeneric is crossing between wheat and rye. That gives birth to triticale. And the second example is cross between carnation and sweet william, which gives birth to Fairchild's mule. So again, now we will see this wide hybridization, that is crossing between the plants belongs to same species, same or different species, belongs to two same genus or two different genera. So for wide development of wide hybrids, there are some barriers. So what are those? First one is failure of zygote formation. Second one is failure of zygote development. Third one is failure of F1 seedling development. So by reading, simply reading, you can say like all three are similar, but no, all three are having some different aspects. Okay. So first one is failure of zygote formation or cross incompatibility. So what is meant by failure of zygote formation? Means when you will dust pollen powder on stigma of female flower, so that pollen is not able to germinate on that particular stigma. So that pollen is not compatible. Okay. So that is why zygote is not formed. So that is one barrier for production of wide hybrids. Then second is failure of zygote development. Though the pollen will germinate, pollen tube will form, but double fertilization will not take place. So as double fertilization will not take place, the zygote will not develop as a viable zygote. Okay. Then third one is failure of F1 seedling development. Failure of F1 seedling development means though zygote is formed, but that zygote becomes sterile. Zygote becomes sterile due to endosperm abortion. As we all know, endosperm is nutritive tissue. Endosperm will provide nutrition to all the seeds which are developing. So after the endosperm abortion, ultimately that seedling will not develop. So all these are the barriers for the production of wide hybrids. As we all know, wide hybridization means crossing between two plants belongs to same or different genus. So because of the genetic genera differences or species differences, this cross incompatibility or hybrid sterility will take place. So there are some techniques by using which we can make this wide crosses successful. So first one is removal of scarification of stigma, so that stigma will become viable and that stigma we can say becomes compatible for foreign pollens. Then second is use of short style parent as a female. Use of short style parent as a female means ultimately no foreign contamination will be there. After the dusting of pollen, no foreign contamination will be there and which will make this wide cross successful. And third one is use of diploid species as a male parent. So after meiosis, haploid gametes can form and which can be useful for doing double fertilization. So what are the reasons for hybrid inviability? So there are different reasons for hybrid inviability. First one is lethal genes. Then second is the genetic disharmony between two parental genomes. As we are taking the plants for crossing, we are taking the plants from different species of same genus or relatively from two different genera. So two different genera means ultimately their genomic composition will be different. Okay. So because of that genomic composition, different genomic composition, their chromosome composition will also be different, and there will be huge gap of chromosome number of one species from one genus and another species from another genus from other genus. So there is a huge gap in for genome purpose also, and as far as number of chromosomes also. So that will make this hybrid inviable. So the third is chromosome elimination. So if we use some species from specific genus where one chromosome either less or one chromosome is more than the cultivated, so ultimately it will result in failure of this hybridization process. So next is incompatible cytoplasm. So as we all know, whenever we'll produce F1 hybrid, the cytoplasm present in the hybrid will come from female parent. So As if the female of that the female used in the hybridization process is not compatible for germination of pollen, and if we will transfer that cytoplasm in the F1 hybrid, so ultimately that hybrid will also become inviable, that you can say as a sterile hybrid. So next is endosperm abortion. Endosperm is the nutritive tissue for developing zygote, developing seed, and developing seedling also. So if the abortion of that endosperm takes place, ultimately this hybrid becomes sterile or inviable. And that will lead to the failure of seedling development due to the complementary action of lethal genes. Then hybrid sterility due to lack of structural homology between chromosome of two species. So as I told you earlier, if we took parents from different species or different genus, so ultimately their genome combination will be different. And if the genome combination of species will be different, there is a huge gap in number of chromosomes. So if there is a huge gap in number of chromosomes, so it is quite a tedious job to make this cross successful in between those two species. So there are some techniques for production of wide hybrids. So first one is choice of parents. So you have to select those parents only where the crosses should be successful and both the parents should be compatible for germination of pollens. Then second is you have to do pollination on sufficiently large number of flowers. You have to pollinate sufficiently large number of flowers so that you can get ample amount of seeds. Third one is reciprocal cross. So as we know, parent A cross with parent B is equal to F1. This is a straight cross. So for making wide hybrid successful, you have to attempt reciprocal also. Parent B cross with parent A for yielding of F1 hybrid. If there are any barriers during crossing, so you have to determine that barrier and you have to take a control measure to overcome it immediately. The next is bridge crossing. Bridge crossing means suppose you have decided to cross between A and B, but B is not compatible with A, but B is compatible with C, and A is also compatible with C. So what you have to do? You have to transfer the desirable characters from A to B via C. First, you have to transfer the characters from A to C, and then you have to transfer from C to B. That means parent C is working as a bridge between A and B. That's why the name given bridge crossing. So next is use of pollen mixtures. Then manipulation of pistils. Then you have to attempt large number of crosses to get the successful wide hybrids. Then ninth is use of growth regulators. So sometimes the stigma will not be viable in some flowers. So you have to use the growth regulators like indole acetic acid, naphthalene acetic acid, and gibberellic acid to make the stigma more viable for specific pollens. Then you can use the tissue culture techniques like protoplast fusion, embryo culture, and grafting. What are the applications of wide hybridization? Align addition line, then align substitution line, then introgression of genes. As we have already discussed, transfer of different characters from different source in particular variety. The characters may be for disease resistant, wide adaptation, quality, male sterility, incompatibility, and complex characters like yield. You can develop new crop species by using this wide hybridization. You can utilize the commercially available hybrid varieties for crop improvement, which is already having a desirable characters. So you can use those F1s to improve your current breeding program. So what are the limitations? What are the limitations of wide hybridization? Okay, so we will start again. Next is limitations of wide hybridization. As we have already discussed, incompatible crosses, F1 sterility, problems in creating new species, lack of homology between chromosomes of parental species, as parental species belongs to different species or different genus. So undesirable linkage. So what is undesirable linkage? While transferring some desirable traits from one variety to other, some undesirable traits will also come. So that is due to presence of linkage. So problem in transfer of recessive genes as due to differences in chromosome homology. Then lack of flowering in F1, dormancy, and problem in using improved varieties in hybridization. So here we can see characteristics of hybridized plants like high resistance, high yielding capacity, high productivity, lifelongness. Example is brinjal, you can say okra, chili, rice, and tomato. So last we will deal with procedure of hybridization. So first you have to set up your objective. What is the objective of hybridization? Why you are doing hybridization? If your goal is development of high yielding resistant variety, then you need to select one of the parent that is high yielding and another one is resistant to a particular disease of your interest. So you have to select the parents according to your objective. So if your parents, if you don't have data related to your parents, then you have to evaluate those parents to check whether these parents to ensure their homozygosity. So before hybridization, please ensure that the parents you are using are complete homozygous. You have to do emasculation. After emasculation, you have to bag. So after 2 to 3 days, you have to bag, and then the bags are removed 2 to 3 days after pollination. Similarly, during bagging, you have to do tagging. Then pollination. Then you have to harvest the F1 seeds. So cross seeds are harvested carefully and stored to raise the next F1 generation. Okay, in case of combination breeding and transgressive breeding, you have to raise F2 population and appropriate solution program you have to undertake. So though the seeds you have harvested, you have to store in appropriate containers with a proper label. So what is the significance of this hybridization? So you can develop the new plants. You can transfer the one or more desirable characters like yield, resistant to some pests and diseases into a single variety. You can produce hybrids which are better adapted to changes in the environment. Okay. And you can use this hybrid, you can attain, or else you can achieve this goal of hybridization by using tissue culture techniques like protoplast fusion. Okay. Hope you have got all these points related to hybridization. Thank you. Thank you very much.